Hello everyone, hope you all are doing well. So today we are going to discuss the next copy of our Aspire for Agriculture test reach. Aspire for Agriculture test reach. So in the previous video we have discussed the section A of test 6. Section A of test 6. So today we are going to discuss the section B of this test 6. In section A we had the parts of soil and water conservation. In section B we will have parts of plant breeding. So you will see the questions of plant breeding predominantly in section B. So let's see what were the questions and how the student has written the answers. And I will be just discussing the overall structure of the answers, and I will not read everything line by line because it will take a lot of time. So I will discussing the structure, important points, intro, conclusion, all the things which I will feel is important, which should be highlighted to you. I will highlight. And if you want to read all the answers, okay, by line, you can simply pause the video and read it. It is on you. Wherever you want to read, you can simply pause and read it. So let's start. The first question which was given was, discuss the genetic consequences of self and cross pollination. So we all know what self-pollination is, what cross-pollination is. Self-pollination when the stigma and anther of the flower of same plant, that is the pollen grains of same plant, is pollinated in the same plant only. And in cross-pollinations, the pollen grain of one plant is pollinated with the other plant. You all know it. Now we'll see the genetic consequences of self-pollination. Obviously, there will be rapid increase in homozygosity. Obviously, homozygous, as we all know, that self replicating plants or self pollinating plants are homozygous and homogeneous, so there will be increase there. They are homozygous, obviously develops homozygous plants. Inbreeding depression can be seen after some generations. Less vital offsprings can be seen. Narrow adaptation can be seen. Inferior seed quality after some generations. True to type plants. So written points, you can simply pause and read it. Now we will see the genetic consequences of cross pollination. Obviously, there was homozygosity, here is heterozygosity. Heterozygosity will be increased. There will be genetic variation. Outbreeding can be caused. They will be more vigorous and vital. Adaptation will be wide. Enhances gene pool. Production of different types of plant, and like parent plant, here we will diversity in production. Diversity, better quality of seed sometimes, and sometimes not always better quality of seeds, but generally you will see in cross pollination there is better quality of seeds due to the features of both the plants. But if both the plants in any one plant is of not desired quality, the seed quality will also reduce. That should be kept in mind. So this was the first question. The second question is, discuss the importance of conservation of plant genetic resource. So we all know what PGRs are. They are diverse collection of plant species and their genetic material. So we all know that there are diverse plants in our earth, and their genetic material leads to plant genetic resources. What are its importance? Obviously, it provides building blocks for breeding crops. It maintains agrobiodiversity, very important, very good point. Ensures future agricultural production, help traditional farming, support ecosystem functions, enhances resilience, support sustainable agriculture and food security. The very valuable points. You can simply pause and read it if you want to read in detail. And well concluded. Okay. Here about 14th convocation of OUIT has been mentioned, although it is a bit specific, but it is okay, where President Draupadi Murmu was present and he discussed something about these things. So well written. And we and now let's move to the next question. The next question is, what is pure line, and discuss the applications of pure line selection. So what pure line selection is? So what is pure line is? Okay. Mhm. So first of all, you can see that the student has missed pure line. What is pure line should be written here. The student has mentioned about pure line selection, but I think a small definition of pure line in one line can be written here. Applications have been mentioned, such as isolation of desirable traits, development of uniform varieties, development of superior hybrids or superior varieties used in parent hybridization. So written points. Now let's move to the next question. The next question is, discuss the approaches of transfer of two or more characters in a single recurrent parent. So how can we do this? So we will see the approaches. Backcross breeding can be done here, obviously. But the student has mentioned only about backcross breeding that needs to be improved. Other features can be written here. But fairly written, the backcross breeding has been well mentioned. Now let's move to the next question. The next question is discuss the usefulness of somatic hybridization. So we all know what somatic hybridization is. Is a technique which involves fusion of protoplasts to form hybrid cell and develop a hybrid plant from the cell. Okay. So basically here we fuse protoplasts to form hybrid cells. So what are its application? Obviously production of new species, heterozygotic crop varieties can be produced here. Production of plants through asexual method can be done here. It can be useful in overcoming sexual incompatibility or self incompatibility. Okay, it can ease the interspecific and intergeneric breeding, help in transferring recombinant organelles. So, well, 10 points. You can simply pause and read it. Now, let's move to the next question. The next question is what is embryo rescue? Discuss its advantages. Okay, so what is embryo rescue? The embryo rescue is a technique that acts as a savior for the revival of immature or weak embryos and then regain this viability. Okay, so you can, the name suggests embryo rescue. That is, we have to rescue the weak embryos. So you can simply get the idea from name only. Then you can write here with the student has mentioned. Factors have been mentioned. Media, we have to take the proper media. Generally, the Cooks media, MS media you all know about. Generally, we take that MS media. Temperature should be well maintained. The light should be well there. Photo period should be maintained. Time of culture has been mentioned. Now, procedure has been also mentioned, such as embryo culture, ovule culture, ovary culture. So, all these things you can do embryo rescue. Now, the main thing is advantages of embryo rescue. Now, see what a student has done. If you look at the question is embryo rescue and discuss its advantages. So, although you have written factors and other things, I am not questioning it, but it should be brief and the more focus should be given to advantages and what about embryo rescue. So I think okay, embryo rescue is fairly written, but equal importance of advantage should be given to advantage. And you can see the student has mentioned all the things, factors and types, but advantages are very briefly explained, which were exclusively asked in question. So you should keep these things in mind. What are the keywords in question and what you need to highlight in your answers. That is why you should keep these things in mind. So advantages of embryo rescue. It is useful in breeding of incompatible species, shortening the breeding cycle, recovering hybrid embryos. So at 10 points you can see producing sterile seeds. So you can simply pause and read it. Only 10 points, but it should be explained. A good value to this part in the question should be given. The next question is and describe in detail the positive and negative negatives of GM food. Okay, so here we have to discuss the positive and negative of negative impacts of GM food. So we will see what GM food is. You all know that genetic modified food which we get from some modification of genetics, some adding of genetic material from other species to get the desired results. Making has been mentioned. Genetic engineering is used. Okay. Now we'll see positive impact. Resistant against disease example, BT brinjal. Generally, BT brinjal is not resistance against disease. You can be here writing about resistance against disease and pest, such as BT brinjal and BT cotton. Both are resistant towards pest. Reducing use of pesticides. Obviously, if BT cotton is used, there will be resistance to pest. Then how will you use pesticides? Improved survival, improved yield, biofortification, such as golden rice rich in vitamin A, or you can say beta carotene. Beta carotene, improved appearance. Okay, the well written points. You can simply pause and read it. Now we will see the negatives. These were the all positives. You can simply pause and read it. Allergen free nuts. So you all know there are various positives of GM crops, which I think you should all know. Now let's move to the negatives. So what are the negatives of GM foods? Obviously, various many times what happens, resistance in pests develops, such as bollworm resistance against BT cotton, which is seen in some cases. Development of allergies, obviously, may cause cancer, due to some mutational changes. Here, instead of cancer, you should be first writing what mutational changes. And in example, you should write cancer. Increased use of herbicide, and you can side write here herbicide resistance, which is seen in the case of glyphosate. Okay, risky to pollinators. Obviously, due to pollination, some kind of new species could be developed, which can be dangerous to our environment and biodiversity. So, bulletin points: risk of extinction, that is dangerous for our biodiversity. You can simply pause and read it. And you can see in conclusion, GESC, that is Genetic Engineering Appraisal Committee, has been mentioned, which is established through Environmental Protection Act of 1986. So, well, it in conclusion. Now let's move to the next question. The next question is differentiate between plant variety and plant variety protection. Sorry, plant patents and plant variety protection. Okay. So what is patents? So it protect a specific. Look, in difference, the student has gone from the paragraph approach, which is good only. But for the convenience of examiner, you should try to write in this box format, because it will be convenient to examiner, and they will fetch you some good marks. I am not saying that there will be bad marks in this format, but from my point of view, this should be most preferable format to be writing differentiate between. Okay, specific DNA, whole set of plants. So writing points, you can simply pause and read it. Examples have been mentioned, such as kesar, mango, pest resistant, brinjal. These are all given through patents. Now, what is plant variety protection? It protects the plant variety. Here you can see it protects a specific DNA sequence. Okay. Suppose there is a variety of brinjal. It is not protected, but here the kind of feature in brinjal, such as it is disease resistance, it is protected here. But in variety protection, the whole variety is protected. Duration have been mentioned, such as 15 years for field crops, 18 years for trees and vines. I think patents is for 10 years. You should check it. So written points, you can simply pause and read it. Here the acts, such as Plant Patents Act of 1930 and PPVNFR, that is Protection of Plant Varieties and Farmers' Right Act of 2001, have been highlighted. So written points, and I think you get to understand various structures in the questions, and from this student mistake you have get to learn. That is why I discuss these types of answer copies, because that could be very beneficial for you in writing and improving your answer writing journey. So that is all for today. If you like it, please press the like button. Please do share, and please do subscribe. Like, share, and subscribe. It takes a lot of hard work to make such videos for you on regular basis. And if you like, if you share, if you subscribe, it motivates us to make more such videos for you regularly. So that is all for today. Have a nice day. Thank you.